So good morning, everyone. Welcome to this webinar. This is part of the, this is hosted by the Statistics and Trials Special Interest Group, or ACTA-STING. And this is the first in a series. Can we have the next slide, please? We'd like to start by acknowledging the traditional custodians of the land on which we meet today and pay respect to their elders past, present and emerging. And we extend the respect to Aboriginal and Torres Strait Island people here today. I'm on the land of the Wurundjeri people. Please, the next slide. And then please ensure that your microphone is on mute. Raise your hand to speak in the Q&A session. You can also use the chat function to put down any questions and we'll get to them. There will be a recording available later on the website and the YouTube channel, and we would also appreciate a post-webinar survey if you could complete that, please. Next one. So we've decided to run a series on DSMB workshops because there's more and more trials that require DSMBs, and we realize that people are asking for information about that. And this is the idea of this three-part webinar we're hosting. Next slide, please. So there are 3 sessions. This is the second one. The first one, the recording is available on the website if you've missed it in person. Today's will be about the challenges of stopping trials for benefit. And then there's one next week on the role of a DSMB in adaptive Bayesian platform trials. Next slide, please. I won't cover the summary of the presentation. I'll leave the presenter to do that. And then just to say thank you very much to Dr. Lehane Tabane, who's doing the talk today. He is the Vice President of Research at St. John's Healthcare in Hamilton in Canada and Scientific Director of its Research Institute and a Professor of Biostatistics at McMaster University. We appreciate your insights and your time to do this. Thank you very much. And I'll leave it to Lehane now.
Fantastic. Thanks, Aneka. Can you still hear me?
I can, yes.
All right. Now, can you see my slides?
Not yet.
Not yet, but things are a bit slow, right?
All right. So I assume Natalie is working on this.
No, so I need you to go back to what we did before because now I can see you, not the slides.
Oh, you can see me, not the slides. Okay. All right. So I have to then go into presenter mode. And then once I'm here, hang on a minute.
You start the presentation at the 3 dots.
3 dots. And then I show presenter view. And then once I'm here, what do I remember?
Then you share in Teams.
Then I have to go into Teams to share. So now I have to find how to get out of this to be able to. Bear with me.
Right. We had this working 5 minutes ago, I promise.
Yeah, it was working. And
so PowerPoint, do the presenter screen.
Yeah,
and then go
screen
into
and
Teams.
then show taskbar, and then go here and share my screen. And I'm going to show presenter mode.
Yes.
All right. How's that? Do you see my presenter mode?
Yes, it is.
Do you see one screen or 2 screens?
No, we see the wrong one.
See the wrong one. Okay, now
But if
taskbar, stop sharing, and then I should share the screen. Show presenter mode. How's that? You see my screen now?
Yes.
That's
yes.
perfect.
Is it moving?
Thank you.
Yes, it is.
Yes.
All
thank
right.
you.
Okay.
Wonderful. Thank you.
Beautiful. Thank you,
Thanks
guys.
for everybody.
Welcome, everybody. Sorry, we've been struggling with the technology, but I think Aneka's husband has been incredibly helpful to get us sorted out. I have a word missing in my title. Is the challenges of stopping trials early for benefit. I didn't realize this until Aneka started reading it. And I'm going to talk about the immediate kangaroo mother care trial as an example, and I'll come to this in a minute why I chose this particular one. I do have no conflicts of interest to declare, but I do get credit for giving presentations like this from my institution. Now, so the last time what I shared with you was about interim data sharing by the DSMBs, what to share, with whom, and why. And today, what I'm going to talk to you about is something that I presented at a meeting last year of hot topics in neonatology. And this was a meeting in Washington. It happens every December, around the beginning of December, and I'm going to do it again this year. These are two of my wonderful colleagues. Both are retired neonatologists who we've continued to work together. They are the ones who got me involved in this movement. And the idea of hot topics in neonatology is about having, hot methodological issues presented at this meeting, really in the form of educational presentations to practicing neonatologists in the field of clinical trials. And both Barbara and Harish have got me involved in this, and I've been doing this on an annual basis for quite some time now. So we were involved in two topics last year. It was about Bayesian analysis of clinical trials, and then stopping trials early for benefit, not for harm. And I'll come to this issue in a minute. And this were some of our co-presenters. And we also had Helga, who was one of the lead investigators of the immediate kangaroo mother trial care trial. So I'll come to our discussions in a minute. So Connie is one of my colleagues here at McMaster who co-chaired this session with me. And then Helga presented the issues about the trial, and I covered the statistical aspects of it, about what the challenges are about stopping trials early for benefit. And then Danielle, who's one of our former trainees here at McMaster, actually looked at an overview of how kangaroo mother care looks like in the US and so on. Now, the session was not about criticizing the immediate kangaroo mother care trial. It was really about using it as an opportunity to learn about issues related to robustness of evidence and issues related to stopping trials early, and how these issues can really become problematic at times. And we only use this trial as a convenient examples to actually discuss these issues. And you'll see as I go through this presentation. Some of the things that you may not be familiar with that I want to highlight at the moment before we get started is stopping trials early for benefit, which is very distinct from stopping trials early for harm. And we can also look at issues related to risk of bias when we do these things. I'll also talk about the number of events, which is an important element in evaluating the certainty of evidence, particularly if trials are stopped early for benefit. And then ultimately, I'll talk about issues of susceptibility to bias, and I'll talk briefly about the concept of fragility index, which becomes essential when we assess whether a trial has potential for risk to bias. So let me give a bit of background. So 15% of neonates worldwide are born preterm, and they account about 70% of all neonatal deaths around the world. And in 2022, there are about 2.3 million neonatal deaths in the world. And if you look at it, 70% is really more than 1.6 million deaths. And unfortunately, the majority of these deaths occur in low and middle income countries in Asia and Sub-Saharan Africa. Now, which is a bit unfortunate when you think about United Nations Sustainable Development Goals, one of their targets is to reduce this neonatal mortality. And they've set a very lofty target of bringing it down to 12 deaths per 100,000 live births in all countries by 2030. Now, kangaroo mother care, I don't need to probably explain it, which is really a type of newborn care of you involves skin to skin contact with the caregiver or a mother or a father to reduce mortality in infants that are born with low weight or preterm to some extent. Unfortunately, the majority of these deaths occur before a baby is stabilized, while kangaroo mother care is often provided after the preterm baby had been stabilized. So the question that the immediate kangaroo mother care trial was trying to look at was, could this intervention be initiated prior to stabilization of the newborn? So they looked at preterm babies. The intervention was kangaroo mother care prior to stabilization. You compared usual care, which is kangaroo mother care after stabilization, because it's now a global standard of care for preterm babies. And the outcomes were mortality at 28 days or 72 hours of age, and the time frame was within 28 days of age, of course. And there was some other outcomes of interest, which we'll come to in a minute. So the trial had an independent data safety monitoring board of 7 individuals, all having expertise in clinical trials, statistics, newborn care, including ethics, particularly in resource-limited settings. And they had 2 planned interim analyses when 50% and 75% of enrollment had been completed. And they had early stopping rules, which was pedo of alpha of 0.001, and this was for benefit. They did actually have potential stopping rule for harm of alpha of 0.05. I should mention that it's often quite rare for trials to actually have a stopping rule for harm, but this one actually did, because nobody really wants to harm babies. So the kangaroo trial showed that actually this thing was doing more good than harm. So there was about 25% reduction in mortality at 28 days. And this was highly significant, and it crossed that stopping boundary when they did the interim analysis. There's were subgroup analysis that were done at that time, and they showed no significant subgroups either by birth weight, gestational age, type of delivery, whether it was vaginal or cesarean, whether it was singleton, twins, size for gestational age, whether it was small or not small, including country. And there were about 5 countries that were involved, 4 from Africa and 1 in India. So they looked at other outcomes, and it shows that actually there was significant reduction, not only in mortality at 28 days, but reduction in sepsis and sepsis-related mortality as well. So WHO was the sponsor of the trial, and they put a lot of weight behind dissemination of the results of the trial. And as a result, the evidence was taken up very quickly to update the guidelines in 2022, with WHO recommending that immediate kangaroo mother care should be done as soon as the baby is born. They shouldn't wait until stabilization has taken place. So that now has become the standard of care. There have been 2 other trials on this issue, I should mention. One was in Uganda. This trial in Uganda actually did not show any significant reduction in mortality at 28 days. This wasn't actually the primary outcome, it was a secondary outcome. The primary outcome was at 7 days. But if we focus on mortality at 28 days, there was no reduction in mortality. Another trial was done in Gambia, and that trial was also not significant, looking at all-cause mortality. So you might argue that, well, we don't often make or change guidelines on the basis of a single trial. We rely on the collective wisdom from different trials, looking at all the evidence together. And if you put together all these three trials, it actually shows reduction in mortality of about 20%, and which is still significant. So if you look at all these three trials, there's definitely trend going towards reduction in mortality. And if you look at the totality, the amount of weight from the trial that was stopped early, it was about 60%. So keep that in mind as we go through this. The key message is, so two other trials could not replicate the result from that trial that was stopped early, and which was a result of 38 mortality that led to the trial being stopped early based on the stopping rule. And of course, if you look at the collective evidence from all the three trials, the message is, hey, there's still benefit at 28-day mortality, as shown by the results of the meta-analysis of the three trials. But there was some concern. People worried that, pooling the meta-analysis, even though that's what we often do in evidence-based medicine, in this particular case, the meta-analysis was done in a way that actually ignored the potential bias from the trial that actually had 60% of the weight, and it is a trial that was stopped early. And I'll come to the issue of why people worried about this. So this raised lots of concerns about the debate. They said, the evidence is dominated by a single potentially biased trial that was stopped early. And I'll come to the issues of why trials that are stopped early, there's evidence to show that we should worry about them. Others argue that, hey, listen, there's no harm in putting a baby on your chest, early or not early, especially because there's some evidence, whether it's from a single trial that was stopped early or trials that are put together in a meta-analysis, that you're saving lives and you're not harming any baby. Others argue that the intervention is less likely to cause harm, and therefore we shouldn't be worrying about this. We should be adopting this evidence, this intervention wide around the world. Okay, what do we know about stopping trials early for benefit? There's two sources of evidence that seems to be consistent in showing that there's a higher risk of overestimation of benefit. It doesn't surprise that people were worried about the results of that single trial or the results of the meta-analysis that included that trial that provided 60% of the evidence. And there's two sources of evidence that shows that there's a high risk of overestimation. It's empirical evidence coming from systematic reviews of trials that were stopped early for benefit. We also have evidence coming from simulation or theoretical studies, and I'll show both of them. Let's look at empirical evidence that seems to show a high risk of overestimation. This was a study done by colleagues here at McMaster a few years ago, and they looked at trials that were stopped early for benefit, looking at trials that were not stopped early for benefit, and they showed that the results from these trials that were stopped early for benefit had implausibly large treatment effects, especially when the number of events were small. I'll come to the issue of how small is small. Now, they looked at this issue later on, another group related to this effort, and they actually again showed that truncated trials that were stopped early were associated with greater effect sizes compared to those that were not stopped early. And it turned out actually that this issue of bias was more related to really the number of events. And it showed that if the number of events was smaller, in other words, they had less than 500 events, the problem was even more attenuated than if the events were more than 500. Keep the number 500 in mind. I'll come back to it. So what about, so in short, empirical evidence showed that there's consistently high overestimation of benefit compared to not stopping early. It turned out actually simulation studies and theoretical studies showed the same thing. So empirical evidence seems to be consistent with what simulation and theoretical studies seem to show. So this was a study that actually looked at this issue when that empirical study was published, and they actually showed that actually this issue may not be as bad as requiring 500 events. You could still get away with overestimation, or if you have more than 250 events. So this was from looking at a simulation study. And then earlier on, Stuart Pocock and his colleague had actually showed that theoretically, there's issues to worry for trials that were stopped. That seemed to also exaggerate the magnitude of treatment effects for benefit, but this was from a theoretical point of view, and which was also supported by the study that was done by one of our colleagues, Stephen Walter, with some of our colleagues here at McMaster. So if you look at the evidence from simulation and empirical studies, they seem to suggest the same thing. There's a high risk of bias for stopping early for benefit. But if you look at the collective evidence from both simulation study, empirical studies, theoretical studies, two critical points seem to emerge. That if you are below 250 consistently, the risk of bias is very high. Between 250 and 500, maybe you might argue that it's medium. But clearly, if you're above 500, the empirical evidence seems to suggest that you are in the safer zone. Now, there's some caveats. So when this information came out, people looked at the evidence from simulation study, empirical study, theoretical study, and they worried that their studies actually looked at the evidence from trials that were stopped early for benefit, and these were trials that were stopped because there was statistical significance, and they were using statistical stopping rules to make that decision. And they were compared with trials that were allowed to continue until the end, but those trials included two types of trials. Some that become statistically significant, some that do not show statistical significance. So these comparisons didn't seem fair, that you're comparing trials that were stopped early, all of which are statistically significant, with trials that were not stopped early, which include two types of trials, those that are statistically significant and those that are not statistically significant. So people argue that this is not a fair comparison. You're comparing apples and oranges. So that seems to make sense. So what happened was that then there was another study that looked at this differently. They only focused on trials that were statistically significant. Others were stopped early, others were not stopped early. And they actually showed that, if you actually stopped when you had 50% or more information, the impact was negligible. So in other words, really, if you did apple-to-apple comparisons, the issue perhaps may not have been as bad. There was still bias, but you had to be above 50% or greater information size for you to be able to do that. So the takeaway message is really stopping trials early. There seems to be consistent evidence from empirical studies and simulation theoretical studies that we run a very high risk of overestimation. But if you compare apples to apples, that seems to say that really the risk of overestimation or bias is not as bad, provided, of course, we stop at the time when we had 50% or more information at that time. All right, so let's take what we know about stopping trials early for benefit, and now let's look at how the immediate kangaroo trial could actually be evaluated in the presence of what we know. So the question we ask ourselves is, do we have enough events that the certainty of evidence for benefit in this particular case could be trusted? So I'm going to share information about three things. One, looking at the first worry, which is bias. The second, looking at precision of the estimate of effect, which is what we often also use when we evaluate the certainty of evidence using the GRADE evaluation criteria. And then I'll talk about fragility aspect, which is another issue which helps us to be able to assess whether the evidence is trustworthy or not. So how many events were available at the time when the trial was stopped? It was actually 440 events. So this showed that, could one worry that the risk of bias is high? In fact, if you look at 440 events, it's less than 500. It falls in that medium risk of bias range. And if one used that as a criteria to judge the risk of bias, you might actually say perhaps for this particular trial at the time when it was stopped, there was a medium risk of bias. So remember again, it was 140 events. But, even if we argue that this is just one piece of evidence, we still have other trials that if we combine with, even though they were not statistically significant, then maybe that risk is not that bad. However, remember that I also said earlier on that the meta-analysis that was presented did not adjust for that risk of bias. So keep that in mind. How precise is the result or the evidence from this trial? Now, this is where we rely on the GRADE platform to be able to look at the certainty of evidence. GRADE, for those who might not know, stands for Grading of Recommendations, Assessment, Development, and Evaluation. This is one of those McMaster inventions which has been endorsed by many organizations around the world. And it looks at really evaluating the quality of the body of evidence on any pickle question and looking at the strength of the recommendation on the basis of that evidence. So if you have randomized controlled trials, really you start with the highest and you then go down depending on whether you think there's risk of bias or the risk is high, low, or serious, and so on. And you look at other issues related to consistency of the evidence and so on. So imagine if you have highest rather than on the basis of the risk of bias, you would grade down. So the issue then becomes, how do you actually assess whether there's a risk of precision in this particular case? The way the grade approaches this is they actually use the benchmark of information size. So the way information size works is, suppose I ran a single trial and I wanted to be able to see how many events would I need to be able to actually answer this question definitively and have confidence that I've got the answer right. So it's really the information size of that trial. And in this particular case, if you're looking for information size, it's over about 630 events, right? Now, remember, if information size is 360 events, we got about 440 events from the trial that were observed. And therefore, the way you evaluate the precision using the grade is you then compare the observed number of events with the information size that would have been required for the trial. Then you realize that 440 is less than 331, and therefore one could argue that perhaps there's a medium risk of imprecision. Then you would grade down the grade rating. How fragile is the results of this trial? This is one of our colleagues, Mike Walsh. Mike Walsh was one of our trainees a few years ago, and he introduced the concept of the fragility of a trial. And this is, suppose you have a trial in which you're comparing two treatments, A and B, and you're comparing them on the basis of the number of events in the two arms. And if you are able to show that the two arms are statistically different in terms of the number of events, and suppose you do this on the basis of Fisher's exact test, you get a p-value less than 0.05. Now, the fragility of that trial would be if I move the number of events in the treatment of interest, which is A, from no event to event until I reverse the statistical significance. That number of events that I need to move from no event to event to reverse the statistical significance is called the fragility of the trial. If I have to move a small number of patients to no event to event, then we say such a trial is less, it's actually more fragile. But if I have to move more of them to reverse the statistical significance, we say it's less fragile. So people looked at this issue when it came out and they applied it in different settings. This was a review of trials in anesthesiology. This was done in orthopedic trials. This was in spine surgery, emergency medicine, stroke. There's another one in stroke. This was ophthalmology. And all the trials reviewed in this area show that many of the trials in these clinical areas are actually incredibly fragile. In other words, it took a very small number of participants from no event to reverse statistical significance. So what is the fragility index for this particular immediate kangaroo mother trial is 21 participants. Now, we don't know whether 21 participants is actually large or small because we really don't have a benchmark for what is the ideal fragility index for a trial. All we know is that the smaller it is, the worse, the more fragile the trial is. The higher the number, the less fragile the trial is. One of my students actually looked at all the reviews that have looked at this issue in most clinical areas, and the average across disciplines was 4. It took 4 participants to actually reverse statistical significance for most of these trials. Now, you can imagine about the evidence or how trustworthy the evidence is that has been used to create guidelines in many of these clinical areas when the trials themselves seem to be so fragile. The take-home message is that really there's greater risk in stopping trials early, and the support came from both empirical and simulation and theoretical studies. And if you look at the immediate kangaroo trial itself, the assessment seems to show that really there's perhaps a medium risk of bias of overestimating the benefit and some medium risk of imprecision if you compare the number of events relative to the information size. And certainly it's hard to know whether it is fragile or not fragile because we don't really have a reliable benchmark against which to assess that. But most importantly, to remember that science is based on replication. We do something in one patient, we replicate that in several patients, we call that a study. But we know a single study is not good enough for us to actually make guidelines or policies on. We rely on replications of the same study in various settings. Now, 2 other trials that tried to replicate that result were not successful. So we have 1 trial that was successful, the other 2 were not successful. Now, this inappropriate stopping trials for benefit, it's just not an issue of, perhaps overestimating the benefit. It could also have, serious negative effects in practice. There's Gordon Guyatt, and perhaps one of the most well-known colleagues in evidence-based medicine who actually came up with the term evidence-based medicine. He's still here at McMaster and works with us. And I spent a lot of time looking at these issues, and many of the evidence I showed you, he was involved in some of those studies, whether it was empirical or even simulation. Many of the people who led those studies were his trainees. So I encourage you to look at his paper, which he also did with some of his former trainees and colleagues. And he actually was trying to illustrate that this is not just an issue of overestimating benefit. It could have serious negative impact on the health of patients. And he came up with 3 examples where there was overwhelming evidence to suggest that people rushed to judgment once the evidence became available, and some trials were stopped early in beta blockers in non-cardiac surgery and intensive insulin therapy in critically ill patients and so on. And some of these things really, they adopted in practice and guidelines, and later on people realized actually they had made a mistake. Now you can think about how many lives had already been affected by adopting interventions in practice where the evidence really was shaky at the time. So they actually strongly recommend that we should stop rushing to judgment on the basis of trials stopped early for benefit. Rather, we should use a very stringent rule of more than 500 events to stop early. That was the argument that they put forward. But they do acknowledge that regardless, it takes time for practice to change. And sometimes, people are doing these things in areas where there's absolutely no evidence. And it's not that simple. And even once something has been adopted in practice and we find that actually we made mistakes, it also takes time to actually reverse that practice. Now, So what is my general advice to people is to say, hey, you know what? We do have an obligation as scientists to also try the best we can to try and estimate benefits well and correctly. But we also have empirical, ethical imperative to try and minimize, the risks to participants who are trying to help us to generate that evidence and so on. So this is really a difficult thing because you're always trying to balance both perspectives. You have to consider the pros and cons of stopping early for benefit. The scientific value for doing it, is that if you don't want to do it early, you run the risk of overestimation. There's potential low imprecision by stopping early, and certainly there's the risk of low certainty of evidence. Replication could be a problem, and you could have uncritical uptake of the evidence, as Gordon has showed with a few examples. So we could have unintended harm by doing so. Yeah, sure. There could be reduction in cost and so on, but certainly it also makes it a lot harder for people to try and replicate a result where somebody has already shown that something works and this trial was stopped early. Imagine people trying to actually participate in another trial of immediate kangaroo trial where the risk of death for this preterms is high. I doubt if anybody would want to participate in such a trial. And I'll come to this discussion in a minute. So the pros is that, if something really works, stopping early can help us to adopt it in practice and perhaps save more lives. Certainly, there's no ethical challenge in keeping participants on less beneficial therapies once the evidence is there. And of course, the DSMB have an obligation to protect participants. So, we don't want to continue exposing people to less efficacious trial interventions once the evidence seems to be overwhelming that something is better. But we also have researchers' obligation to use equipoise to justify randomization. And if the evidence is such that we could no longer feel comfortable that we can randomize individuals, then perhaps one has to reconsider. But I often say it's better to be realistic and be pragmatic and realize that, stopping trial early for benefit it's complicated. That's why you need both clinical and statistical considerations and people who have expertise in both areas for you to be able to do it. There is no perfect solution. It's a big dilemma. I often encourage people, I think I talked about this, is to recruit seasoned scientists to actually help you by saving on your DSMB and agree on the stopping rules, which is not only statistical criteria, but the number of events. Information size is incredibly useful, and make sure that these things are documented in the protocol and in the charter for the DSMB as well. You could actually say, you know what, I'm going to adopt Gordon's advice and say, don't stop trials early. And, that's one extreme. Or you could say, well, stop when we have at least 50% of the information. That seems to show that the risk of bias is perhaps not as bad. Or you could say, have 50% of the information in, and you also need more than 500 events, which is one of the criteria that was shown from empirical studies that Gordon recommended. Or you could say, well, we need 50% and use much more stringent stopping rules that PHRI. This is Salim Youssef at the bottom, and Shrikant Bandiwala, who is a statistician for the PHRI group here at McMaster. They use a much more stringent rule, and I'll talk about this in a minute. PHRI is one of our institutes here that has done very, very large pragmatic trials around the world in cardiovascular trials. And they have a very large global print in multi-country trials. And they have, taken this issue very seriously and have come up with their own criteria for trying to minimize the risk of stopping trials early for benefit by making the stopping rules much more stringent. And they call them the PHRI boundary rules and so on. I encourage people to have a look at their paper. They've used these rules in many of their trials, and the idea of being simple, trying to take all that we know about stopping trials early for benefit and guard against all the potential challenges that we could face if we stopped early. Or you could say, I'll do a combination of PHRI rules, 50% information, and number of events. So you could use a combination. So I often, share these options with people as my advice to them to say, you could either say, hey, you know what, I'm not going to stop at all, or use any one of these combinations of rules that can help you to guard against inappropriate early stopping. There's additional resources that I would encourage on for you to have a look at in terms of stopping trials early that people have written in a way that I think is helpful to people. So every time I present this, I often actually list, a few things for us to think about, to discuss, and when is it really appropriate to stop trialing. That was really the purpose of presenting this at that meeting, hot topics neonatal area. And what are the associated biases and how can we avoid this? How do we balance the ethics of stopping early versus the pursuit of scientific truth? That's what we're trying to do. The scientific implications of stopping early, the ethical implications of not stopping early, and so on. So I presented this at the Society for Clinical Trials last year. Was it last year, or it was earlier this year actually, in Vancouver? And we discussed all these issues. There's Susan Ellenbeck, who spent a lot of time actually on issues related to DSMBs and stopping rules. And Susan was in the audience as well as we're talking about this. And I've worked with Susan on several things. And he says, you know what, for me, I really wouldn't care. We should be stopping a trial if there's evidence that the trial is saving lives. The most important thing is still saving lives. And I think it would be hard to argue against Susan when you think about it, because who wants to harm babies, let alone preterm babies that are so fragile in the first place and so vulnerable? But there's also worry about this attitude that, yeah, we want to save lives, but what if we are trying to actually pursue saving lives at the risk of getting it wrong? So it becomes difficult. I doubt, honestly, if anybody's going to try to replicate that trial, because with WHO behind it, guidelines already changed. I doubt if anybody is going to have the courage to try and change this. So what about the biases in the meta-analysis? You said, hey, listen, we have methods of correcting for that bias. The problem is people don't use these methods. I'm just putting up this papers on the methods for people to be aware of them, that really we do have methods for correcting for bias studies when we do systematic reviews of trials. But unfortunately, most people don't use these methods. So the other problem with them is that there's no simulation studies telling you which method to use. Any one of those 5 methods could lead you with different answers. So the issue is which one do you take, which is what Janet Wittes, who is also one of the experienced trialists who served on several DSMB, will say, yeah, sure, but which method would I use if all of them give me 5 different answers, right? So this created lots of discussions. Actually, I should mention that Jared actually served on the DSMB of the recovery trial during COVID. And he said during what they were given, they didn't actually even have a clear stopping rule. They were just told, you guys should recommend stopping when you think there's convincing evidence that will change practice. Mind you, people were dying during COVID, right? So now, there was some clinician in the audience who actually said, I think you guys are asking the wrong question. Yeah. And to them was like, they were taught that you should only stop if a reasonable individual would agree that stopping is the right thing to do. So the issue then becomes, how do you define reasonable? Would everybody agree on the criteria for reasonable? So he thought, that's the question we should be asking. And this is Tim Ramsey, one of our colleagues at Ottawa Health Research Institute. And, he was just admitting that, this is not an easy thing. It is quite a challenge for PIs of trials and DSMBs. There's usually no easy answers to it. And then there was somebody in the audience who said, a lot depends on the context and the disease. There are areas where actually there's nothing that works. And if people seem to find something where the evidence suggests that you could actually save lives, then perhaps it might actually be good to stop early in that particular case. Susan Halaby works in cardiovascular trials. as you were saying, hang on a minute, guys. You are saying we should only stop when we have 500 events? That's not what we do in cancer. And quite a lot often in cancer trials are stopped early because, hey, people are dying and the trials are very small. And so they don't have the luxury of going on until they get 500 events. So you can see how difficult this issue is, and it could vary from context to context. So I'm hoping that I've given you the gist of the challenges about this issue, with the hope that perhaps we can find ways to work around it. I will stop here, Aneka, and pass it on to you for questions.
Thank you very much. I guess we could stop the screen share if we could see everyone. Maybe my question is, also in the example you gave, we talk about stopping trials early for efficacy, but your efficacy endpoint is also a safety endpoint. So if we decide to not stop early for efficacy, but to stop if there's a safety signal, isn't that forcing us to stop early for efficacy, if I make sense?
Yeah, I mean, mortality is usually a very difficult thing to deal with, which is quite often the case where we actually even recommend that people should have a DSMB whenever mortality is used as an outcome, because of the dual nature of mortality. Yeah, one could argue that yes, you're thinking of it as benefit, but it's also safety issue, right? Which makes the discussion even more challenging, especially when it comes to babies, right?
Yeah. I don't know, Natalie, can you see questions? I cannot see any questions. Does anybody have a question they want to ask in person?
I don't see any questions in the chat. I don't see any questions in the Q&A.
One's just come in from Sabine. In the beginning, you mentioned a stopping rule for safety in the trial. Could you elaborate on this rule in the trial?
Yeah, so they put the issue of safety. I can't forget what outcomes they actually worried about safety, and it was 0.05. But it wasn't really used as a basis for stopping the trial. It was on the basis of using the 0.001 stopping rule for benefit that was used, not so much that safety issue. It was a very low threshold for safety, to be honest, not for benefit.
Lots of great presentation and thank yous. Any further questions? Jennifer's just come through with one. If I'm understanding this correctly, events refer to a dichotomous outcome. What about continuous outcome measures?
Very good point. Yeah, the context of the use of events is for trials that use binary outcomes. We don't have analogous rules in terms of events for continuous outcomes, but the principles of stopping rule in terms of the rule itself as based on a p-value could still apply. The only problem is you don't have the number of events, you don't have information size for you to be able to do that. So information size is often on the basis of the number of events as well. So quite often, or to be honest, when we worry about trials, we often are worrying about trials that have binary outcomes, especially when we are monitoring safety or monitoring efficacy. typically trials that use continuous outcomes are not that large, because using a continuous outcome is going to reduce the number of participants you need for a trial anyway. We worry about trials that require a lot of information to answer the question, where by the time we get the answers, we may have killed so many people unnecessarily.
Thank you. So if Natalie has shared the slides, we can just do our final few announcements. And if anybody types a question while I'm doing the final announcement, we can still answer that question. We just want to advertise some of the things Sting is doing. One is our DSMB mentoring program. It's already closed for this year, but just so that people are aware of this, you could apply in future. We'll be matching the mentees and the mentors really soon, and you can find more information about this on the website if you'd like to see, if you're interested. Next slide. We also remind you of the clinical trials and registry symposium that's held later this year. Registration is still open if people are interested in going. And then next slide. Thank you, and please do the questionnaire we'll send you. And also a reminder that there's the webinar next week on the DSMBs and adaptive trials. And there is one more question from Sabine. You mentioned the PHRI stopping rules, and how are they different from the classical rules, for example, Habertal?
Yeah, so in the presentation, I do have the actual publication on the PHRI rules. I encourage you to actually get access to it. And you'll notice when you look at the rules that they're much, much more stringent than the well-known classical rules. So they're just much more stringent. I haven't seen any other more stringent rules in the literature than PHRI stringent rules, to be honest.
Thank you very much. And thanks again for your time and for everybody attending. Thank you.
Thank you, guys. And thank you for inviting me to be part of this.
Thanks.
Bye-bye for now.